drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Seyfried, thank you for joining me.
Well, thank you very much. It's a pleasure to be here, Nick.
Can you start off by just telling everyone a little bit about who you are and what your background is?
Yeah, thank you. I'm a professor of biology at Boston College in Chestnut Hill, Massachusetts. I currently teach cancer metabolism. Cancer is a metabolic disease. I teach general biology to the non-majors, going over fundamental aspects of evolutionary biology and biochemistry as well. And we focus on maintaining or improving scientific literacy, which seems to be in very short supply among members of our society. So we try to improve understanding of becoming more scientifically literate and being able to interpret information. So I teach those classes, and I've been here at BC for almost 49 years, 40 years actually, and pretty much running my own research program for all these years on a variety of different backgrounds: epilepsy, lipid biochemistry, cancer metabolism, and now predominantly cancer metabolism.
Mm-hmm. And let's just start out with some basics around cancer. Can you just give us a good definition for what cancer is? What characterizes cancer cells as opposed to other cells? And what I'm interested in here, besides the definition too, is do you have a definition that differs at all from what we might call the orthodox definition you might find in a textbook?
No, I think the definition in the textbooks are what everybody would agree upon,
Hmm.
and that definition is cell division out of control
Hmm.
or dysregulated cell growth. The controversies and the discussions are on what is the origin of that and how you manage that.
Hmm.
So, but the definition is pretty well accepted throughout the scientific communities. Cell division out of control, dysregulated cell growth is what is generally referred to as cancer.
Hmm. So cell cycle has been disrupted somehow. The cells are growing in a dysregulated fashion. There's no disagreement on that. That is what cancer is. It's pretty intuitive too. I think the average person gets that, well enough. But there's definitely different ideas out there around what's causing that dysregulation to begin with.
Yeah, that's right. That's where the main discussions and controversies are today as to what is the origin of the dysregulated cell growth. How does that happen? What would induce a population of cells in a particular organ to begin a proliferation that is no longer regulated?
Hmm.
We do in our normal cells, normal tissues, we have cells that will divide, grow, and replace other cells that have been worn out and degraded from age and wear and tear. Probably the biggest or one of the most interesting aspects of normal cell division is regenerating liver. A lobe of the liver can be taken off either an animal or a human, and you can get massive, rapid regrowth and replace a large number of cells that were removed in the lobe. And that's rapid cell division under control. It's not dysregulated control. The cells know when to stop. They don't continue to grow, as opposed to a hepatoma, which is a cancer of one of the cells in the liver that has dysregulated growth. Those cells they no longer respond to the inhibitory signals that the cells in a regenerating liver would have. So we have cell divisions that occur naturally in different organs, the gut, the Crypt cells of the gut. All of these normal cells are dividing. They have cell division, but in contrast to cancer, those cell divisions are under control, and where cancer cells are out of control.
Mhm. So out of control or dysregulated doesn't merely mean that the cells are growing quickly, because as you said, we've got cells that divide quickly in different parts of the body. That happens, but it's happening in a regulated fashion. They know when to grow, they know how fast to grow, and even if they're growing quickly, it's done as it's supposed to be done. It's done in a regulated fashion, and it's only when somehow, some way, the cell cycle controls on that division get disrupted that things become cancerous.
Yes, that's right.
And so when I remember very clearly being a biology student all the way back in high school, college, and so forth. I never studied cancer in the lab. So my exposure was just in the textbooks and in the classroom. But I remember very clearly being told that, we sort of knew how cancer arose. And that is, well, we've got the genome. The genome contains the full suite of genes. Every cell's got a genome inside of it, the nuclear DNA. And there are tumor suppressor genes and there are oncogenes. So these are genes that encode things that somehow, some way are tied into the regulation of cell cycle. And when you acquire multiple mutations in certain genes, oncogenes promoting cancer, tumor suppressor genes suppressing cancer, if you get some combination of mutations in these types of genes, then you will get cancer. And that is how we get cancer. That was what I learned. And to what extent is that true for certain cancers? And to what extent was that just sort of a one model that I learned about as if it was the model?
Yeah, well, I learned the same model, and I read the same textbooks, and I believed. I had no reason to challenge it. That what you just described as the model, the tumor suppressor genes, the oncogenes, and all these kinds of things, because I had taught genetics. I have a degree in genetics from the University of Illinois, and we were all led to believe that. I didn't I was a mammalian geneticist, and we looked at Mendelian inheritance of different things. And then we learned that, cancer cells have a lot of mutations, and they seem to be out of control, driven by what you just said, tumor suppressor gene defects and oncogene turn on and all this stuff. And we had to became deeper and deeper into the kinds of signaling cascades and the phosphorylation, dephosphorylations, and all these kinds of things that we get more and more in depth on this. It wasn't until I started look when we work on epilepsy. We mapped genes for epilepsy, many different genes, and we studied this for many decades at Yale University when I was there, and here at Boston College. And we were always finding new genes, but we never were able to manage the epileptic seizures with the information that those gene mutations were telling us. And then we learned about this ketogenic diet that seemed to work to manage epileptic seizures, regardless of the origin, whether it was a genetic-based seizure, whether it was acquired-based seizure, viral infection seizure. There's a lot of ways you can get epileptic seizures, but it seemed as though this ketogenic diet was able to manage a broad range of seizure disorders. And then we said, well, what's the common mechanism by which this diet could manage these different seizures of different origins? And it was never clear to us.
So can I just, so is one way of sort of saying all of that, if you go back in time and then you come back forward in time, we've got the molecular revolution, we have the Human Genome Project. We start to figure out like, okay, here's the genome sequence, here's the gene for this, the gene for that. There was this thinking that there was going to be a gene for everything. And so as an example, in the epilepsy field, you're saying people started to uncover genes that were associated in some fashion with epilepsy. And as we accumulated that knowledge, it didn't actually tell us how to solve epilepsy. So we had all these genes that were related to epilepsy, but it wasn't really telling us what to do treatment-wise. But then you had this diet, the ketogenic diet, which was effective for treating many forms of epilepsy, and it did not depend actually on which mutations you had or didn't have.
Yeah, and it was kind of surprising. And then we were doing angiogenic research on brain tumors here. We had found that there were certain glycolipids that were provocative to angiogenesis. And at the turn of the century, around 1999, 2000, the work in cancer was the angiogenesis surge, the abnormal vascularization of tumors. Mm-hmm. And Judah Volkmann had come out from Harvard saying that if you have an antibody, you could shut down against vascular endothelial growth factor. And, Jim Watson of the Watson and Crick DNA fame said, Judah Volkmann has found the cure to cancer by targeting blood vessels and shrinking the tumors down to nothing. So we were studying angiogenesis from the perspective of glycobiology. And we were very excited about finding certain glycolipids that actually had an impact negatively on angiogenesis. But then we were also doing calorie restriction for epilepsy, and then we found out that the work from Peyton Rouse about 100 years ago showed that calorie restriction could shut down angiogenesis. So we were saying, well, what's going on with the calorie restriction? And then we found out that the calorie restriction could also manage epileptic seizures. The ketogenic diets given in restricted amounts managed epileptic seizures by lowering blood sugar and elevating ketones, and calorie restriction did the same thing. And then, of course, we started seeing calorie restriction really shrink angiogenesis in cancer cells, and it was linked to the level of blood sugar in the bloodstream. So we had published a paper showing there was a direct relationship between how high your blood sugar was, and these were in mice. We did linear regression analysis, how fast the tumors would grow, and how the blood vessels would be reduced in conjunction with lowering blood sugar. So I said, wow, this is really interesting. Anybody ever seen anything like this before? Of course, Otto Warburg had seen it a long time ago.
Okay, perfect. Because I was going to ask you about some of the history here, because I'm not a cancer guy, so I don't know all of the details here on the history. But as I started to read your stuff and other stuff in this realm, it wasn't that, one thought I had is, okay, well, we thought that the somatic mutation theory was the way things worked, that you accumulated these mutations in tumor suppression oncogenes. And that does appear to be the way it works, at least for some cancers to some extent. But then, over time, our knowledge has grown. But then I started reading some of your reviews and I realized, oh, no, actually, for a long time, there's been these different competing ideas on oncogenesis and how cancer arises. There's a deeper history here than I realized. So who's this Warburg character and what are his ideas?
Yeah, well, I didn't know who he was. In science, we all hear of the Warburg apparatus and Otto Warburg. His name had always been kicked around in the background of whatever we did. He had a major impact on biochemistry, one of the leading biochemists of the 20th century. And he was involved in a lot of different kinds of things, the photosynthesis. Um, he had produced many. Not only did he have a Nobel Prize, but Hans Krebs worked with him. He worked with a lot of leaders in the field of biochemistry. So I started to think bigger about, like, well, who the hell is this Warburg? What did he say and how did he say it? And then why was his ideas shelved? Why did the field move away from him onto what we call the genetic idea of what cancer is, with tumor suppressor genes and oncogenes and all this kind of stuff? So I started to go back, and my research on mice with brain tumors and metastatic cancer, controlling it by lowering glucose and elevating ketones, showed me a power that rarely was seen in a lot of the genetic ideas about cancer, with respect to improved quality of progression-free and overall survival was really quite improved significantly. And then I said, well, what was it that Warburg said that was inconsistent with what the worldview was? Otto Warburg was an interesting character. He came from a history of scientists, physicists, and this kind. And he was trained in the rigid, quantitative Germanic way of doing science. And he claimed that 1 mole of oxygen consumed equals 7 moles of ATP produced, and that 1 mole of lactic acid produced equal 1 mole of ATP. And he was able to devise a Meyerhof quotient unto determination of oxygen consumption related to ATP production. And immediately he ran into a buzzsaw with his own work, as well as that of Sidney Winehouse, who at that time was the director of the National Cancer Institute after the Second World War. And Winehouse came right out and said Warburg's hypothesis is not correct. Even though many cancer cells consume less oxygen than normal cells, Warburg attributed that to a disturbance in oxidative phosphorylation, leading to a compensatory fermentation. So in other words, as cancer cells lost their ability to generate energy through oxidative phosphorylation, they were forced to compensate the lost energy with the fermentation mechanism, which is energy without oxygen.
The idea there, for those that are not well versed in this, so our cells can make energy using our mitochondria and something called oxidative phosphorylation, which requires oxygen. And that's a really good way to make a lot of ATP, a lot of energy. There are other ways to make energy and they're less efficient. The idea here that I think it sounds like you were articulating for what Warburg was talking about was, okay, if oxidative phosphorylation is compromised, the cell will shift to using something like glycolysis, a less efficient way of making energy, because something has disrupted this other way it has to make energy.
Yes, and glycolysis would be super upregulated in cancer cells relative to normal cells, and he felt that massive upregulation of glycolysis, which is what they call lactic acid fermentation, was the signature of the origin of that was the disruption of oxidative phosphorylation, and he said if you disrupt oxidative phosphorylation too acutely, cells will die, because they will not have an opportunity to compensate. So a chronic disruption of oxidative phosphorylation led to a protracted increase in in fermentation, lactic acid fermentation.
Hmm.
And then he was trying to quantify those phenomena by showing that cancer cells took in less oxygen, but they produced a far greater amount of lactic acid, showing the balance, that the overall energy balance was compensated for by a non-oxidative energy metabolism. And that was his big thing. That what Sidney Winehouse jumped all over him on was that, which leads today, which many folks in the field today are still behind Sidney Winehouse, which we have eviscerated completely, but also because of Warburg made some mistakes. But the bottom line here is that Warburg had found a couple of tumors that actually were consuming oxygen at the same rate as normal cells. And Winehouse pointed those out and said, how can you possibly have an insufficiency of oxidative phosphorylation when you have oxygen consumption equal to that, the normal cells and tumor cells?
Yeah.
And Warburg said, well, the fermentation was unable to put out the oxphos. The oxphos and the cells was unable to squelch the fermentation.
Yeah. So I guess
others, others then jumped on the idea that, oh no, this is a problem with fermentation. It's an abnormal fermentation, has nothing to do with an insufficient oxidative phosphorylation. And the field jumped on this because oncogenes and all this became the drivers of the fermentation. And going back to the oxygen consumption argument of Warburg, it was wrong.
So as an episode in history, this is perhaps a good lesson because, biology is messy. Our knowledge is incomplete. Warburg had ideas. Other people had ideas. There were exceptions that seemed to contradict some people's ideas, and people were debating, why are we observing this if this is true, and so on and so forth. And so the score was never really settled there. People disagreed, but it sounds like what ended up happening is the field sort of agreed that one set of ideas was true, even though things weren't truly resolved.
Yeah, and I think the biochemists were beating each other up over these observations, and everybody coming out with a slightly different interpretation. Was Warburg right? Was Warburg wrong? What does this support him? Did this not support him? And that was all during the 1930s and 40s and 50s. And then, of course, with the Watson and Crick Nobel Prize for the structure of DNA, and of course many others were part of this DNA revolution. And back at the early part of the century, in 1914, Theodore Boveri claimed that cancer might be due to some sort of chromosomal aberration, and maybe it's the chromosomes that might be, because every time you look at a tumor cell, there was always abnormal chromosomes in there, and that was just more or less a kind of an off. And Boveri himself, clear in that first, he said, it was all speculative. He said, I have no knowledge. He's never studied cancer in his life. He said, I'm embarrassing the field. I'm an interloper. I don't know what I'm talking about. He said, the cancer could be just as due to something in the cytoplasm, but I have no idea. I'm just speculating on this. Anyway, when Watson and Crick came out, oh, wow, this genomic stuff may have been already known way back in 1914 by Theodore Boveri. Oh, my God. So we anointed him as the father of the gene theory of cancer, Theodore Boveri. In the meantime, biologists have always been inferior in some ways to physics. Physics is the pure intellectual idea of human brain. The highest level of quantitative assessment comes from physics. So many biologists have always had physics envy. And when Watson and Crick came out with the quantitative analysis of nucleic acid patterns to link to amino acid structures, we now bring biology into a molecular range, far more quantitative and less messy, as you said.
Yeah. It becomes seen as a harder science.
Harder, absolutely. Even the physicists were jumping into the biology area now because they felt that they could use some of their skills and knowledge to further understand the molecular biology of the cell. and why it was just like a stampede of people. Everybody, you had to do something molecular. Biology was no longer a
Hmm. Hmm.
systems approach.
Yeah.
It was now a molecular kind of
And one thing I think I want to emphasize here, and I'll, get your input if you think this is true. You know, science is this thing that people have invented, and we have this sort of ideal of science, what it looks like if you're fully rational and dispassionate and you're making totally objective observations. But we have to remember also that it's a human enterprise and humans are social creatures, and we all have careers and aspirations and motivations that have to do with the social realm. And once the molecular revolution happened in the mid 20th century through to the Human Genome Project, and really until very recently, in my opinion, one of the ways that you could make a real career for yourself as a scientist in the molecular biology realm was identifying a gene for something, identifying an oncogene or a tumor suppressor gene. And so there was this really, really sort of simplistic gene centric mindset developed because you could really make a whole career on cloning and isolating and identifying genes involved with cancer or some other disease. And it got us into this sort of very simplistic gene centric mindset, I think. Is that how you see it, or how do you see it?
I think it started that way for sure, in a less complicated way, which was thought only to explode into a hopelessly complicated situation, which we have today.
Mm-hmm.
But before we got there, we thought that we could identify specific mutations that could be responsible for the dysregulated cell growth. And then it became as the field evolved and Warburg was left behind. And getting back to your human nature behavior, Sidney Winehouse had some animosity towards Otto Warburg because of Hitler sparing him during the Second World War, and he should have been gassed during the Holocaust. Why was Hitler sparing him? And the stories that Sam Apple brought out in his book Ravenous, what Hitler feared cancer like there was no tomorrow. His mother died of breast cancer or something. And he knew Warburg was the guy in the world that would have a better understanding of cancer than anyone. And if there was going to be a solution to this cancer problem, it would have been Warburg. So he survived Second World War working with, he was no fan of Hitler, believe me. He threw the brown shirts out of his lab. I mean, he was quite, but he just wanted to continue with his research. And then after the war, Winehouse and other Jewish folks were concerned that why did he survive when the rest of us had to suffer?
I see. So they had an interpretation that maybe there was some nefarious reason for that, even though apparently there wasn't. And remind me, Winehouse was at the National Institutes of Health at a pretty high position. So that was a politically powerful position that probably enabled Winehouse to have their ideas spread further and faster than they otherwise would have been.
Yeah, and the other thing too is Dean Burke was a strong supporter of Warburg, and Dean Burke was also before Winehouse the head of the National Cancer Institute, and he was thrown under the bus with Warburg. For them, we're discounted for various reasons, and because the field was so much more excited in the gene. And getting into the molecular basis for this, and felt that the guys in the biochemistry were not really doing anything. And in 1976, Winehouse wrote this paper about Warburg's flicker of metabolism, flickers only dimly now as the field has moved away from this ambiguity. And he eviscerated Warburg, and he even said to himself, he says, "I hate to." Say unfavorable things to a guy who died. Warburg died in 1970, and Winehouse wrote this paper in 1976, and pretty much, just ripped apart everything that Warburg had said and why he was wrong and all this kind of stuff.
So we've got the science actually happening. People are in the lab, they're making observations, they're coming up with hypotheses, but there's also these political and social career human aspects to what's going on. So people have, political motivations or career motivations to go with or against other people. They want certain ideas to spread, others not to spread. And so, that's all mixed together with legitimate observational and scientific disagreements that were happening.
Absolutely. That's the way science works. You have an idea, and if you can get enough support for your idea, you can turn the direction of the field. And as the genes, more and more genes became recognized in cancer cells, I think there were Nobel prizes for oncogenes and tumor suppressor genes and these kinds of things. And then it gradually became more and more clear as the technology for genetic analysis improved over the years, that cancer cells had thousands and different kinds of mutations. How is it possible that any one of these mutations could be linked to dysregulated cell growth? So then we had to parse the types of mutations that were present in these cells, and we called them at least Dr. Vogelstein from Johns Hopkins started to parse them out into passenger genes and driver genes. So the driver genes became more recognized as those that were more likely to be involved in dysregulated cell growth than some of these other passenger genes. And then it became clearer and clearer that we couldn't find specific drivers that might be responsible for dysregulated cell growth, like the p53 tumor suppressor gene. What was thought to be the key driver of cancer, but not all cancers had mutations in p53, and it was not always so clear about what was going on. So then he introduced the dark matter. Maybe these the real drivers are in this called genomic dark matter that was nowhere to be found, or nobody knew what it was. But it was saying, the dark matter. So what does that mean? It started to become more bizarre as it was not possible to specifically identify a particular gene or even a group of genes that could account for the dysregulated growth.
Yeah,
so
this is a common.
So let me just say, when we started to take apart cancers and we started to separate them into individual cells within a tumor, and then clone up those cells and then do genomic screens on the individual cells to get a better picture, every cell in the tumor, not one cell had the same genetic problems as any other cell within the same tumor.
Yeah, and so this is another common pattern in science, I think, where someone has an idea, they have a hypothesis. Naturally, you want your hypothesis to be right. You want your theory to be correct. So you're trying to gain support for it. But very often, you start to accumulate these observations that just don't fit with it, things you can't quite explain. And what often happens is that the hypothesis just becomes more and more elaborate, and you start adding all these auxiliary hypotheses to your theory, and you come up with these new concepts. This is like what you meant by the dark matter concept and stuff like that. You have to make your theory more elaborate to accommodate the observations that actually aren't quite fitting with the original idea. And before the theory actually dies or goes away, sometimes years or decades can go by where it becomes this sort of complex and elaborate monstrosity compared to what it was originally. And it's because you're starting to accumulate ideas over time that don't actually quite fit with that idea.
Yeah, well, that's exactly right. And that shows the almost exactly what you said was the situation for the geocentric theory of the solar system. For 1,800 years from the time of Aristotle to Ptolemy, it was thought that the Earth was the center of the solar system, and the mathematics to explain the movement of the planets became more and more complex with epicycles, deferents, and equants, and it turned into a giant massive ball of thread, challenging all of the minds at that time to come up with better and more sophisticated mathematical formulas to explain the movements of the planets. Even Copernicus himself was struggling with these same mathematics until he put the sun in the center of the solar system using the same mathematics. Now you could predict. You became predictable as to where the planets would be, and the sun was the center of the solar system, with the Earth just simply being another planet moving around along with the other planets, and everything called the Copernican revolution, and everything made sense from a massive amount of complexity, and right now in the gene theory, we're stuck in this quagmire of more and more complexity, like you've just mentioned, ignoring facts that have emerged also in this complexity that are very inconsistent with the somatic mutation theory. For example, Sir Michael Stratton out of England, Sir Michael Stratton, for his massive work he's done on genomic revolutions, genomic analysis, he published a paper in breast cancer showing a significant number of breast tumors that had no mutations. Using the same technology where he found other breast tumors loaded with mutations, he found some about 30% of breast tumors that had no mutations. And I said, wow, this is interesting. What does he say about it? And the answer was nothing. It was not even mentioned. The data were presented, but there was no discussion
He just
about
kind of didn't talk about it.
No, didn't talk about it. And then there was a paper by Parsons and a big group. Can't remember where they were. Maybe Johns Hopkins. I think they were at Johns Hopkins as well, where they did a big screen on glioblastoma, and they looked at about 25 or 30 cases, and they found mutations in all of the major mutations in the PI3 AKT signaling, the three major signaling pathways, except one sample. It had no mutations in any of the major pathways. The B20, in their figure 3 of the paper, and the BR20 sample had no mutations. And I said, "Wow, how do you explain that?" And of course, not a single word was mentioned about the single sample that had no mutations in any of the 3 pathways. We're now seeing more cancers. There's some ependymoblastomas in children where they have no mutations.
Yeah.
Yet they all have dysregulated cell growth. So that was kind of like, whoa, a head scratcher, because those findings are not consistent with the somatic mutation theory. And now with the deeper sequencings over the last few years, we're seeing driver gene mutations, P53, as well as others, with significant mutations in the structure of the P53 and other driver mutations in normal tissues, in normal cells and tissues all throughout our body, with no dysregulated cell growth associated with the mutations in the driver genes.
Okay, so you're saying over time we've accumulated these weird examples where in some cases you have cancer cells, but they don't have mutations in the oncogenes or the tumor suppressor genes. And in other cases, you've got normal cells that are not cancer cells, and they do have those mutations. And so these things have just sort of accumulated to some extent, at least, and they largely went sort of unexplained. People just sort of said, well, it's just one cell line in that paper, and they just really didn't talk about it.
Well, and some of the more recent papers have come out even discussing the cancer cells that have normal cells that have driver mutations, and they don't really like to discuss the fact that there's some cancer cells that don't have mutations, because they say, well, you can't support anything by the absence of evidence. Maybe our techniques weren't as sophisticated. There's always some excuse, but it's harder to explain the driver mutations with mutations in normal tissues. Yeah. So what they say is we now have to parse the concept of driver mutations and say that the search is not yet complete for some super category of driver mutations that we seem to be missing, even though we've cataloged over 100 million different kinds of mutations. We still have not found the ones that leading to the dysregulated cell growth. So the search is not yet complete.
Yeah. So the two ways to view this conundrum are, okay, the search is not complete, the mutations must be there, and we just haven't found them yet. Or the alternative would be, well, you're looking up the wrong tree here.
Yeah. Well, that's hard. That, as you mentioned at the very beginning, Nick, it's very hard to say you might not have been correct in the theory that you built your career around.
Yeah. Okay. So
let me say one more thing. The most devastating evidence against the somatic mutation theory has come from the nuclear mitochondrial transfer experiments, where the nucleus of a tumor cell is now placed in a cancer in a normal cytoplasm.
Wow. So you would expect the normal cell to turn into a cancer cell because it's going to now have all of those mutations that are supposedly there.
Yeah. So and you don't get that. And the Israel Schaefer experiments, along with in vivo Rudy Yanish's group down here at MIT, we had Jim Morgan's group at St. Jude's. We had Beatrice Mintz at Thomas Jefferson. All of these classical developmental biologists were finding evidence that was inconsistent with the somatic mutation theory. So you pile that on top of the some cancers without mutations, normal cells with driver mutations, and a cancer nucleus with supposedly the drivers and the tumor suppressors placed into a normal cytoplasm with normal mitochondria, is the neoplastic phenotype is completely suppressed. And on the other hand, if you take the normal nucleus and put it into a tumor cytoplasm, you end up with cells that are dysregulated in their growth.
Wow.
These observations clearly state that the origin of dysregulated growth resides in the cytoplasm, not in the nucleus.
Wow. Okay. So literally people can do experiments where you've got cancer cells, you've got non-cancer cells. You can suck the nucleus with all of the nuclear DNA out of a cancer cell and put it into a normal cell. And if it's the case that the cancer is caused by mutations in that nuclear DNA, you should then cause the non-cancer cell to become a cancer cell because you're putting in all those mutations, but you don't see that. And then you're saying vice versa, you can take the nucleus of a normal cancer cell or non-cancer cell, so it's got none of these supposed mutations in it, put it into a cancer cell, and it's still a cancer cell. And you're saying that indicates that whatever's causing the cancer to be cancer is in the cytoplasm rather than the nucleus.
Yeah, absolutely. And those discussions of the nuclear transfer experiments very interesting how they respond to them. One person, a leader in the field, one of the most recognized leaders in the field, was confronted with the data with those mitochondrial nuclear transfer experiments, and said, if those experiments are true, the whole cancer field needs to be turned upside down and be recognized as incorrect. Therefore, those experiments cannot be true. That
Wow.
preserves the dogma of what's going on. In other words, they're so profound in their interpretation that it can't be true. Yet the leaders in the field of developmental biology were the ones who did this. And the beautiful thing about that, they were unbiased. They were not asking whether cancer were a genetic disease or a mitochondrial metabolic disease. They were simply asking the question, can the nucleus of a tumor cell direct normal development? And the answer is yes, up to a degree. And then the system would abort. But they never thought, I was the one who took all that and I reinterpreted the data in light of the somatic mutation theory, that this is completely inconsistent with the somatic mutation theory of cancer. Those guys were not asking that question. They wanted to know how far in development a cancer how far could a cancer nucleus drive normal developmental processes. And then it turns out that there are mutations in the nucleus of the tumor cell, but it seemed to be the products of those genes were absolutely essential for moving the organism from one developmental epoch to the next. And when that gene was not there, the system imploded and aborted. And that's what they said. That's what they found.
Okay. So in essence, you've got two competing schools of thought: the somatic mutation theory, which is basically that you get DNA mutations and that drives cancer. You've just laid out some of the history there, some of the problems that kept popping up with that hypothesis. You've also got this other school of thought, what some people have called, what you've written, the mitochondrial metabolic theory. And this is more akin to Warburg's ideas that look at it as more of a metabolic and mitochondrial issue. So if I go back to what you said about Warburg's ideas, tell me if this is a fair characterization of sort of what the essence of his hypothesis was. If you shut off a cell's ability to use oxidative phosphorylation, its mitochondria, to generate energy, if you do that quickly, the cell's going to die because it's just not going to have enough energy to live. However, if you do that slowly, if you sort of in a prolonged fashion choke off the mitochondria's ability to use oxidative phosphorylation, the cell will adapt to that in some sense by using glycolysis and other forms of energy production more than its mitochondria. And that is then going to lead to the formation of cancer. Is that a fair summary of what that idea was?
Yes, and we just have to get into it a little bit more detailed and show the actual processes by which that can happen.
Yeah.
And what happened with Warburg? So he was rigid in saying that 1 mole of oxygen consumed was 7 moles of ATP. And the field was locked into that idea that oxygen consumption is a good marker for production of ATP. We just published a paper very recently using a novel experimental design showing that brain tumor cells, both mouse and human, oxygen consumption is not an accurate marker for ATP production through oxidative phosphorylation.
So
So the field is using
people were making an assumption that there would be a really tight correspondence between the oxygen consumption of a cell and how much it was using oxidative phosphorylation, which sounds like a very natural assumption because oxidative phosphorylation depends on oxygen. But what you're saying is that assumption is actually not that good.
and for a cancer cell. It's very accurate for a normal cell, but not accurate for a cancer cell. And Warburg was basing his, actually, it was Hans Krebs who developed the technique for assuming that, or for clearly showing that oxygen consumption was linked to ATP production. And he did those experiments. And Warburg simply assumed also, like everybody else does, that this is true, that oxygen consumption is a surrogate marker for ATP production. And in normal cells, it is. But in cancer cells, it's not. They're uncoupled. They use oxygen to produce reactive oxygen species with much, much less ATP coming from the oxygen consumed than what would be happening in the normal cell.
Okay.
So we were the first, not only us, there was a few other experiments done in the scientific literature showing that cancer cells use oxygen for activities other than ATP production.
Yeah.
So this was the big mistake. And now today, listen to this. We're using the Seahorse oxygen consumption machine instrument sold by the millions and millions of dollars all over the world for measuring ATP, for measuring using formulas to assume how much ATP is being produced for oxygen consumption, never using the direct chemical measurements like we did.
Yeah.
So the idea is we use bioluminescence showing the degree of ATP. Seahorse instrument that most people use now to say that oxygen consumption is normal in cancer cells. Yes, the cancer cells are taking in oxygen, but they use a formula to estimate ATP. They're actually measuring ATP. We're the only ones, the first group to actually measure simultaneously oxygen consumption with ATP production, and there's a disconnect in cancer cells.
It's I mean, I wish more people outside of science understood how common this is, but when you are using, it's very common in science for something like this to happen where you're using calculation to stand in for direct observation. And because you're doing math and because it seems fancy and rigorous, you lose sight of the fact that you're not actually observing things directly. You're making assumptions that are masquerading as observation.
Yeah, that was one thing. And the other thing that Warburg did not know about, and nor do many people today know about, that the cancer cell generates energy through two different types of fermentation. One is in the cytosolic, in the payoff phase of the glycolysis pathway, in the Embden-Meyerhoff-Barnes pathway, where you get ATP there, and everybody knows that's upregulated in cancer cells. But the problem is there's different types of pyruvate kinase. So you produce a lot of lactic acid, but it's not an accurate marker for how much ATP is coming because the pyruvate kinase M2 isoform allows lactic acid to be produced, but it's not linked to ATP production through that step. So measuring lactic acid is not an accurate marker for determining how much ATP you're getting through fermentation. That's number one. Number two, many people in the cancer field know cancers are glutamine addicted.
What is glutamine?
Glutamine is an amino acid, most abundant amino acid in our body. It's in the bloodstream. It's a very valuable amino acid to run our immune system, our urea cycle. It's needed for nucleic acid synthesis. It's for our gut, the health of our gut. We use glutamine. Glutamine is the most abundant amino acid in the body, and it's there for very specific reasons that we know about. In any event, the field knew that cancer cells were addicted to glutamine. So you put cancer cells in oxygen and you see they're taking in glutamine. Oh, glutamine is being respired. It's called an anaplerotic. I hope I'm not getting too deep in the science here for you.
No. Keep going.
Because it's an anaplerosis. Anaplerosis means the replenishment of the TCA cycle with carbons. So when you look at oxygen consumption, you see oxygen consumption, and the glutamine is pouring into these cancer cells, and they thought the glutamine was being respired. We now have direct evidence that the glutamine is fermented. in the mitochondria by mitochondrial substrate level phosphorylation. So the misinterpretation of the data, because my cancer cells are taking in oxygen and they're blowing out ATP, and we're seeing the ATP. The problem is, what's the origin of the ATP? It's coming through a fermentation glutaminolysis pathway, dumping out succinic acid as a waste product. So succinate does not stay in the TCA cycle; it's get blown out as a waste product. Yeah. So this is all new stuff. This is brand new stuff.
Yeah. So basically, one way to summarize some of that is our cells have multiple ways of creating ATP, the currency of energy in our cells. There's oxidative phosphorylation in the mitochondria. There's fermentation in the cytosol. There's lots of different details that can go into exactly how oxygen and other things like amino acids get sucked in and then converted into energy. And many people have made mistakes or assumptions about, if I measure this one output, it means this is being used or that's being used. And a lot of those assumptions have been mistaken in the past. And so there's been a lot of confusion generated about what means what in terms of inputs and outputs with respect to energy production in the cells.
Yeah, absolutely. And if you go in the textbooks, it says predominantly cytosolic ATP production, fermentation, and oxidative phosphorylation. But there's what we now broken it down into traditional biochemistry. We have two forms of ATP production. One is through oxidative phosphorylation in the mitochondrial inner membrane through a chemoosmotic hypothesis of Peter Mitchell. And then we have substrate level phosphorylation. And substrate level phosphorylation is the movement of a phosphate group to an organic molecule to produce ATP. So you can produce ATP with a non-oxidative process. And there's two of those. One is in the cytoplasm and one is in the mitochondria itself. At the succinyl-CoA ligase reaction, alpha-ketoglutarate to succinyl-CoA to succinate, and you get, everybody said, it's GTP. In cancer, it's ATP. And they make a buttload of it in the tumor metabolism, and they're dumping out succinic acid. Let me give you, people in the heart field, the cardiovascular field knew about this, but never turned it over to the cancer field. For example, when individuals have heart attacks, what you see right after the heart attack is a buildup of lactic acid and succinic acid in the bloodstream. And those are the fermentation fuels that are compensating for the acute loss of oxidative phosphorylation. And your neurons need to have a proton mode of gradient. They need an electrical process across the membrane. Otherwise, they're going to die. They cannot compensate for loss of oxphos chronically to upregulate substrate level phosphorylation. So what happens is that you build up this massive amount of succinic acid and lactic acid, and as soon as the blood oxygen come back in, succinic acid and lactic acid both disappear because they were needed there for a short period of time to maintain the electrical chemical gradients. Otherwise, you're going to be dead. So cancer cells are dumping out lactic acid and succinic acid, even in the presence of oxygen. Very different than what happens during heart attacks and interruptions of acute interruption of oxidative phosphorylation. And this is what Warburg said. Warburg said cancer cells continue to ferment in oxygen, but he didn't know that that glutamine was the second major fermentable fuel, and that caused a lot of confusion. So we've cleared all that up now. It becomes very clear. We've cleared up the ambiguities associated with all these assumptions, and now we have hard data to support how these cancer cells are in fact living in the absence of oxygen using a fermentation or in minimal levels of oxygen.
So a couple of things I want people to get here. So under normal conditions, non-cancer conditions, I'm breathing in air. That oxygen's going into my blood. It's getting to all my cells, and they're using that as an electron acceptor in the electron transport chain of the mitochondria. This is part of the oxidative phosphorylation way that we make a lot of our energy. If I start working out really, really hard, I can't keep up with how far I'm pushing my muscles. And so things shift from, I was sort of running out of oxygen in a sense. And so things shift towards glycolysis and these other forms of energy production. And that's what builds up things like lactic acid. And that's why, say, you start to get tired and your muscles burn when you're working out hard. Is that accurate?
Yes. And you also build up some succinic acid as well.
So what you're saying is, okay, that's how things work under normal conditions. We would assume that if lactic acid is building up and things like this, it must mean the cell's not able to use oxygen. But what you're saying is tied to Warburg's ideas is what actually seems to happen in some cancer cells is they kick out some of these things that you would expect to see in the absence of oxygen, even in the presence of oxygen.
Absolutely. Absolutely. And the big thing now is they're kicking out both, listen to this, they're kicking out lactic acid and succinic acid. Succinic acid leaves the TCA, always should stay in the TCA cycle if you're respiring. But the tumor cells are dumping succinic acid out. And not only that, succinic acid stabilizes HIF-1 alpha, a major oncogene. The stabilization of HIF-1 alpha allows the glycolytic pathway to proceed at maximal efficiency, turned on. So the oncogenes that we have now found out, like HIF-1 alpha and MYC, these oncogenes are facilitators of allowing the cell to shift to a fermentation metabolism. Because if OXPHOS becomes chronically disrupted, oncogenes turn on, open the floodgates to get glucose and glutamine into the cell to replace the reduction in energy through oxidative phosphorylation.
Yeah. Well, I think this starts to bring us to the idea that I wanted to get to, which is, we can juxtapose a somatic mutation theory and a mitochondrial metabolic theory here for oncogenesis, but these things are not mutually exclusive, right? You can have metabolic shifts that say generate reactive oxygen species and lipid peroxides that then mutate the DNA. And as you just mentioned, you can apparently have DNA mutations that actually then facilitate or lock in metabolic shifts.
Well, I don't know if they're mutations. This is what Greg Semenza found when oxygen becomes deprived, HIF-1 alpha, which is made and broken, becomes stabilized, and that immediately upregulates the fermentation pathway. So it's not really a mutation;
it's a gene expression change.
Yeah, it's a gene expression thing. So what happens is that you are now allowing oncogenes to be expressed. And of course, if the oxygen comes back, the oncogenes are downregulated.
I see. So it's metabolic change in tandem with epigenetic changes in expression of genes rather than mutations in the sequence itself.
Absolutely, yes. So and this is what was shown when you take a cancer cell that has oncogene expression. And you have abnormal mitochondria in there, deficient oxidative phosphorylation. You see these oncogenes on, and when you put new mitochondria, the oncogenes turn off because you don't need them anymore because they're respiring. But they're essential to maintain viability of the cell when oxidative phosphorylation is insufficient to maintain viability. So they'll do it.
So one thing I want to ask you about. Because I want people to get this piece because it's important here, but it's also just more generally important when we're thinking about cell biology and metabolism, generally speaking. You mentioned the term coupling or uncoupling. And a lot of, as you just told us over the last few minutes, a lot of confusion in this field, in the cancer field, arose because people didn't understand the relationship between oxygen and ATP production and how that varied, how tightly those things were correlated or not under different conditions. And you said that the cancer cells are often relatively uncoupled. When you say the word coupling and uncoupling with respect to mitochondria, ATP production, and oxygen, what does that mean? What is coupling?
Yeah, well, coupling means that the production of ATP through the F1, F0 ATPase, sometimes they call it complex 5. It's a very elaborate protein complex in the inner membrane of the mitochondria. That proton motive gradient that builds up in the inner membrane space of the mitochondria, the ATP production is coupled to the degree of protons accumulating in the inner space. So those protons come passing back through the F1, F0 linked to the production of ATP with oxygen pulling the electrons through all these different proteins. So oxygen serving as an acceptor of electrons is directly coupled to the production of ATP through the F1, F0 ATPase.
So when this is happening, when things are tightly coupled, oxygen, it's almost like a conveyor belt. And oxygen's the thing that's sort of making the conveyor belt turn to generate the energy. And so you have very, very maximally efficient ATP production when you're in a tightly coupled regime. But then the extent that things become uncoupled, what happens?
Yeah, well, here's the situation. During that when you say that coupling, it's not only the oxygen pulling the electrons through the various protons of the electron transport chain. That pulling of electrons is linked to the pumping of protons.
Mm-hmm.
So the pulling of one pumps the other,
Mm-hmm.
creating the proton motive gradient, which is then coupled to ATP production. It's very an incredibly sophisticated machinery. But when you have uncoupling, the proton motive gradient, the hydrogen protons, pass back through the membrane into the matrix.
They leak.
So you're leaking. Yes, you're leaking. Now some of these leakages can be directed by uncoupling proteins. And some of these uncoupling proteins are actually activated in cancer cells. So you're dissipating the proton motive gradient, the electrochemical gradient, by dissipating that. That's called uncoupling. But it's a very important natural part of normal cells to know how to couple and uncouple the membranes.
So you can go in both directions. It's regulated. Sometimes a cell wants to be more coupled or more uncoupled.
It has to be very carefully regulated because this is the way we produce heat. When we uncouple, we produce heat rather than ATP. So the energy is generated by thermoregulation rather than ATP production. But this in some tissues allows us to do this depending on the environmental situation. So it's a very sophisticated system. But in cancer, it's broken. They're uncoupled and they're broken. They're not able to generate an active proton motive gradient, thereby causing the cell to force into a fermentation. Because as Warburg said, energy is everything. Without ATP, nothing can survive. You must have ATP to allow all the gene expression profiles. You have to have ATP. How is that coming?
So how general is the observation, how firm is the observation that in cancer you tend to be more uncoupled in your mitochondria than coupled?
The evidence for that is the massive production of reactive oxygen species.
So uncoupling tends to produce more reactive oxygen species.
Yes. So you have to be very careful about it. And cancer cells are known to have a massive amount of the hydroxide radical, and those things are carcinogenic and mutagenic. So the DNA mutations that you see in cancer cells, many of them, a lot of them, are caused by the reactive oxygen.
So are you saying, so an implication of that, if I'm hearing this correctly, is that so if mitochondrial uncoupling leads to less efficient energy production and it produces more reactive oxygen species and things that can cause mutations, could it be true that historical observations of oncogenes and mutations, this was actually a downstream effect of this kind of metabolic change that happened rather than being sort of the cause?
Yeah, absolutely. That's it. That's one of the things. And just like I said, with the nuclear transfer experiments,
that would explain why in some cell lines you see these mutations and some you don't. It's because this was all happening downstream of this mitochondrial metabolic change, and the mutations aren't actually necessary, but they might be observed sometimes as a consequence of the reactive oxygen species production.
Yeah, and not only that, also from the acidification of the microenvironment. Because what happens when the cancer cell is not efficient in generating energy through oxidative phosphorylation? Yes, it makes a lot more reactive oxygen species, which further damage the ability of the efficiency of the electron transport chain. But don't forget, what are the waste products of fermentation are lactic acid and succinic acid, which acidify the microenvironment, creating more disturbances in the nuclear genome. So when people look at cancer, they always say the nuclear genome is shot to hell. Wow. And it can be all varied in different ways, in different cells, and different conditions, all very different from each other.
So let me ask you this. This is just something that's coming to mind spontaneously for me. So if all of that is true, then I think it makes a very clear prediction with what I'm about to ask you. So obviously, the extent to which our mitochondria are coupled can change based on environmental conditions. You can become more or less coupled or uncoupled, at any given time. That being said, there are maternal haplotypes, so the type of mitochondrial DNA and mitochondria that you inherit from your mother, that are more intrinsically coupled or uncoupled. Some of us have a mitochondrial haplotype that makes us tend to be very, very coupled, and some of us have one that tends to make us more uncoupled. Is it known whether people with more intrinsically coupled mitochondrial haplotypes tend to get cancer at lower rates?
The answer is I don't know. Okay, I think that question needs to be looked at a little bit more carefully. Now knowing that cancer is a mitochondrial metabolic disorder, that type of question should be looked at in a little bit more serious way.
But you think
Because
that is that a good question or a good way to think about it?
I think it needs to be looked at. I certainly think there needs to be some evaluation of that. But it was never really thought about. If you think the disorder is caused by somatic mutations, your entire academic focus is on something other than that.
Right.
So now that once you realize that there's a mitochondrial metabolic disorder, questions like that now become interesting. Let's go back and see if we can look at that. And we know that yes, the haplotype that those mitochondria are eventually distributed to all different cells and tissues in our body. And they all have different levels of uncoupling as part of their natural environment and what they do in those organs. So some are generated from more heat, others are more capable of coupling than uncoupling, and all of that are the sophistication and modifications of mitochondrial function ending up in different tissues and organs of our body. And so we need to look at that. We know that cancer cells there's substrate level phosphorylation is an absolute essential phenotype for having dysregulated cell growth. And the rarity of cancer in neurons of the brain and in mature cardiomyocytes, they are incapable of shifting to substrate level phosphorylation. So you rarely, if ever, get cancers in cells that cannot ferment. But the level of uncoupling that would allow the variability in cells needs to be looked at more carefully. The most important thing, again, how do we connect this to dysregulated cell growth? Okay, we have all this. It turns out, and very clearly, that the proton motive gradient in the mitochondria through calcium signaling controls the cell cycle. So the cell cycle, how we should divide and how we should become dormant and quiescent, is all under the control of the calcium gradients within the mitochondria. So what happens when that mitochondria loses its capability of producing energy efficiently? The cell falls back on its default state, which is proliferation. All cells, when life first emerged on our planet, there was no oxygen in the atmospheres, yet we had cells that were dividing, and they were dysregulated in their cell growth, out of control. And as Albert Szent-Györgyi said, this alpha period of life on Earth was purely unbridled proliferation driven by fermentation metabolism. There was no oxygen in the atmosphere. So it was only the fusion of the archaeobacteria that was actually able to capture oxygen and then lead to the metazoans, multicellular organisms. Before that, everything was a single cell. So these cancer cells are simply falling back on ancient fermentation pathways that were present on all cells prior to the oxygen coming into the atmosphere.
It's the default state because that literally was what came first from an evolutionary perspective.
Yes, and the default state of cells is proliferation, and the default energy state is substrate level phosphorylation because there was no oxidative phosphorylation. And these cancer cells are falling back, and they will grow out of control as long as they have fuels to ferment. And the two fuels that have been identified, we interrogated all these are glucose and glutamine.
Glucose and glutamine. And so that's a sugar and that's an amino acid. Neither of those obviously is a fatty acid.
No, they can't burn fatty acids. They accumulate lipid droplets in the cytoplasm. So you need a good mitochondria to burn fatty acids. And what happens, you see all these cancer cells loaded with lipid droplets. And those are the fatty acids that can't be metabolized.
I see. Okay, so I was looking at that figure in one of your papers. These big bubbles of fat accumulate in these cancer cells, and you're just saying that's fat that's accumulating because the cell can't actually use it for energy production.
Absolutely, and in fact, if you take cells and put them in hypoxia, they start accumulating lipid droplets, and as soon as the oxygen comes back, the lipid droplets disappear.
Wow. So this is immediately so here's where my mind's going. There's a couple ways we can take this, but here's one way that connects to other podcasts I've done. So I became really interested in lipid peroxidation and how that relates to dietary fat intake. And there's been some interesting observations recently in the literature. People like Tim Yateman and Halliday at the University of South Florida, they have observed that that colon cancer basically seems to be coming from these highly inflammatory states that are closely linked to lipid dysregulation. And what you're telling me is, if you're right, that mitochondrial metabolism gets disrupted. You can't use oxidative phosphorylation as well. You can't oxidize fatty acids as well. You start making more reactive oxygen species. This will increase the odds that you will create lipid peroxides that are toxic to the cell. And, if I go back to the Halliday and Yateman work, if your cells have accumulated a lot of, say, polyunsaturated fatty acids, you've just got more stuff now that can be turned into toxic lipid peroxides from reactive oxygen species. And so I'm starting to connect some dots here for how things in the lipid literature and that stuff relate to oncogenesis based on the mitochondrial issue that you've been talking about.
Yeah, absolutely. And the key lipid in the inner membrane of the mitochondria is cardiolipin, which is polyunsaturated. And you start ROS damaging those, and you start to reduce the energy efficiency of the electron transport chain, the efficiency. So yes, you're going to see a lot of, and you do see all this when you do really careful examinations of lipid like we have done in shotgun lipidomics. We see all kinds of lipid abnormalities in these cancer cells, any one of which could be produced by peroxidations and all these kinds of things. And they just contribute to the inflammatory cascade that's going on in this. And then what happens is you create what we call a wounding. It's like the immune system sees that abnormality as a wound. And they come in and they throw cytokines and growth factors on top of this, which creates then a worse situation. They're doing things out of context, making a bad situation much, much worse. So you can see that. And then we showed how that leads to metastasis because the immune cells start to fuse with these cancer stem cells, leading to abnormal immune cell function, leading to spread of cancer cells around the body, all driven by glucose and glutamine. So the way we know we're right is by the response of patients to targeting glucose and glutamine at the same time by transitioning the whole body over to therapeutic ketosis. The cancer cells can't burn fatty acids or ketone bodies, and they're dependent on glucose and glutamine for their dysregulated growth.
Is that generally true? No cancer can burn fatty acids?
No, because they accumulate them in the lipid droplets. They can't. We have. In fact, we throw it out to the field. Anybody who can show us a cancer cells that can grow on fatty acids or ketones in the absence of fermentable fuels, I'll give them $100. I have the $100 here. Guess what? No, in fact, one of my colleagues said, I'll give you $1,000. The cancer cell can't grow without fermentable fuels. It can't burn. Fatty acids are non-fermentable.
Okay, so this brings us to now the question of, so there's two questions I want to get to. We're going to have to take them separately. So there's the question of, if you put people into ketosis with a ketogenic diet, to what extent can this prevent or treat existing cancers? And then there's the question that we have to get to eventually, which is, if broken energy production in the mitochondria is a cause of cancer, we have to then ask, well, what's breaking the mitochondria to begin with? Let's take those one at a time. So talk a little bit more about ketosis and the use of a ketogenic diet, say, as a cancer prevention or treatment modality.
Well, years ago when I did the calorie restriction stuff, I said, well, when we did the comparative analysis of mouse and human, you have to understand basal metabolic rate. The basal metabolic rate of a mouse is 7 times that of a human. So when we were getting these spectacular responses to calorie restriction in the mouse, we said, wow, let's see, how do we translate that over the human? It's water only fasting. So I said, oh my God, because we saw water only fasting in the human produces the same biochemical parameter changes you see in a mouse under 40% calorie restriction. So we thought, oh, just cut down your food by 40%, you'll get what this mouse does. No. You got to go water only fasting because the differences in basal metabolic rate. So I said, wow, that ain't going to fly. How many people with cancer are going to say, oh, now I got to not eat for 14, 20 days, just drink water? People don't want to do that. So we were working with the ketogenic diet and epilepsy, and the little kids seemed to be okay with that. So we said, why don't we try to, because people, that seems at least you're eating something. You're not like feeling you're going to starve to death. And then we started to work the ketogenic diets, our knowledge of ketogenic diets for epilepsy applied to the cancer problem. And that seemed to be a viable solution until everybody went nuts about eating lard and doing all this crazy stuff with ketogenic diets. But what it actually does is a Paleolithic diet, which we realized can put you into nutritional ketosis if you eat foods that are low in carbohydrates, restricted amounts of highly restricted carbohydrates carbohydrates, you can get into a state of nutritional ketosis, just like our Paleolithic ancestors, where there was no highly processed carbs and foods that would raise blood sugar levels very high. So exercise and low carbohydrates will put you into a state of nutritional ketosis. Now, that state is very, very good for the normal cells of our body because my late friend Richard Veach, Hans Krebs's last student, and I would talk for hours about the value of ketone bodies being a super fuel. They generate more ATP without producing reactive oxygen species. Unbelievable. So we can transition the whole body over to ketones, making the rest of the body super healthy, reducing systemic inflammation, a fuel that the tumor cells can't use. So how do we do that? And the answer is we can take, like on our recent Greek study, you should see this. We were able to keep these glioblastoma patients alive for 3 years or longer on these Mediterranean ketogenic, calorie-restricted Mediterranean diet. It was tremendous. So there's so many ways to get into nutritional ketosis, not just using ketogenic diets. You can use low carbohydrate diets. Your body will start producing ketones naturally from these situations. So that's what we're doing. We're transitioning people to nutritional ketosis, which lowers glucose and replaces lowers blood sugar and replaces the sugar with ketones. The brain can burn it, heart can burn it, muscles can burn it, cancers cannot burn it, and then we have to go after the glutamine targeting with drugs, because we have to target the glutamine with drugs and exercise.
So is the essence of what you're saying here, or part of it, that if you put someone into nutritional ketosis, they're using ketone bodies like BHB to generate energy much more so than they're using glucose, because it's a very ultra low carb diet and you're getting a high fat intake. So is what you're saying that you're essentially, because cancer cells can't use those fatty acids, that you're essentially starving them, but your other non-cancer cells are not starved because they can use those ketone bodies?
Yes, it's a transition. Our body evolved. We're a very versatile machine. We have survived on this planet under the most stringent conditions. We're all alive today because of the flexibility of our bioenergetic systems. The cancer cell have compromised that bioenergetic system by having dysfunctional oxidative phosphorylation, pigeonholing them into a fermentation metabolism.
Yeah. So they're metabolically inflexible, which is the very adaptation that is part of what makes us human in many ways, you could argue.
Yeah, perfectly. Yes. And yet the cancer field says, oh, cancer cells are so flexible. They're so versatile because you're always giving them fermentable fuels. As long as they have fermentable fuels, they'll appear to be very tough.
You said something earlier. So you said the ketones. I want to ask, so sort of pound for pound, if you're giving a cell ketone bodies as opposed to glucose, how much ATP can be produced from the ketone bodies molecule for molecule compared to glucose, and how much reactive oxygen species are kicked out when you use one substrate versus the other?
Okay, our bodies will make ketones in the liver. So you will mobilize the triglycerides out of the adipose cells, and they'll go into, as insulin goes down, glucagon, the hormone that triggers the mobilization of fats at adipocytes, goes up. So our bloodstream starts to fill up with triglycerides. You can see it on the TLC plates. We did it. And those triglycerides then go to the liver. And the liver takes the two glycerol backbones and can make a glucose molecule through gluconeogenesis. But that level of glucose is just residual because the brain will suck up like a vacuum any available glucose. And then it's like putting fatty acids into a wood chipper. The liver chops them up into water-soluble ketone bodies, which now circulate through the bloodstream and go to all tissues that have adequate oxidative phosphorylation. So where are we getting our energy from is a ketone body. Now let's get back to why the ketone body is such a powerful fuel, because they have more carbon-hydrogen bonds than does a pyruvate. And now I'm going to explain this to you. So we get more reducing equivalents, NADH, out of a ketone body than we do out of a pyruvate molecule. So we're producing the fuel for a greater amount of fuel for oxidative phosphorylation by burning a ketone body, number one. Number two, ketone bodies deliver all their electrons to complex 1 with very little being delivered to complex 2. So we create a more oxidized coenzyme Q couple, which leads to an efficiency. That proton motive gradient will go up ever so slightly without they do not uncouple. So ketone bodies do not uncouple.
So you're saying ketones, if you're on a ketogenic diet, your body's using ketones, that promotes greater coupling in the mitochondria.
Yes, a 28% better coupling than pyruvate. Now, people will say, well, why don't you burn fatty acids? Because they have a lot more carbon-hydrogen bonds than does the ketone body or the pyruvate. And what happens is they create a very steep proton motive gradient, almost like then the body has to release the pressure. So they automatically turn on uncoupling proteins naturally. So they dissipate the proton motive gradient, reducing the delta G prime ATP hydrolysis. Even though they have more carbon-hydrogen bonds, it's too steep. It's too steep, and the body, they naturally open up uncoupling, regulated uncoupling proteins, thereby reducing the efficiency of energy with a fatty acid. The ketone body does not uncouple, produces very few oxygen radicals, and has more carbon-hydrogen bond does the pyruvate, thereby calling it a super fuel. For each breath of oxygen you take in, burning ketones, you get more power from that bioenergetic efficiency of a ketone body. This is unbelievable. So this allowed us to survive the most harsh conditions of famine and droughts and all this, because we're able to shift from one fuel source to the other. And our brain is naturally capable of burning ketones. They burn sugar, they burn glucose, and they burn ketone bodies. But we are capable of transitioning our nervous system over to ketone bodies. So that keeps us alive and functional.
So some people argue, well, if you give the human body, glucose and ketones, if you give it carbohydrates and fats, it will preferentially burn the carbohydrates, and therefore that's what our cells want, and that's the better fuel supply. What do you make of that argument?
Well, I think that's true in part, not because it's a better fuel supply, is that when you take in carbohydrates, you turn off glucagon. Insulin goes up, glucagon goes down, and you piss out the ketones. So it's the way the body is utilizing this. You have to look at the hormonal balance. When you're in nutritional ketosis, insulin is low, glucagon is elevated. Yeah. As soon as those carbs come in, insulin goes up, glucagon goes down. You don't need the brain will preferentially burn glucose for sure, but it has the capability of adapting to ketones slowly. That's why you can't go real quick.
Yeah. So I'm going to give you sort of my take on some of this, and I would love to get your reaction to it. So a lot of people love to get into the holy wars of carbohydrates are a better substrate, fatty acids are a better substrate, ketosis is better, carbohydrates are better. My sort of view, when I take an evolutionary lens to look at how humans evolved and the remarkable metabolic flexibility that we have, whether or not one group of humans ate a ketogenic diet or a high carbohydrate diet or whatever, I think in pre-industrial times and Paleolithic times, for most of human evolution, people were transitioning from one metabolic state to another, probably on a seasonal basis at least. And so in some sense, it's not that we're supposed to be in ketosis all the time, we're supposed to be in carb burn all the time, but I think we're probably adapted to switch between these on a seasonal basis and as a function of the feast and famine cycles that naturally happened for most of human history. And so it's actually the metabolic flexibility and the switching between states that might be the thing to focus on here, rather than trying to pigeonhole people as being, we should eat carbohydrates or we should eat fats all the time.
Yeah, you're 100% right on that. We have to look at our evolutionary past and who were we as a species. Yes, we were feast and famine. And during seasonal periods when fruits were ripe and you had more carbohydrate, people and ripe berries and all this kind of stuff, you would gravitate towards that. And that's the beauty of our body. It's a very flexible machine. We evolved to be extremely flexible in how we, during seasonal times, exercise, all this kind of stuff. The tumor cell is not flexible, so it cannot make those transitions readily. If you're locking a body into a low carbohydrate diet and a ketosis, it's going to put pressure on the tumor cell. But then if you switch to a high carbohydrate diet really well, the body knows how to do this, the normal cells, but the tumor cell, even though you'd be giving it the fuel, it can't make the rapid transition. So you can just go up and down as long as you know how to use. This is why we developed the glucose ketone index calculator, which will be the new tool for managing cancer and chronic disease in the population. This is a ratio of circulating glucose to circulating ketones, and it comes out as a ratio. And we know that we exist in certain zones in our life that put us at risk. If we have high GKI all the time, we put ourselves at risk for a broad range of chronic diseases and mitochondrial damage as the result of a variety of insults on the efficiency of oxidative phosphorylation. But if we go down into the green zone, which is a GK of 2.0 and below, we're going to put a world of hurt on these cancer cells. But how long do we stay in the green zone before we flip back up to a yellow? In other words, going up and down, because our bodies, just like you said, Nick, we evolved to be in these different states seasonally. Where the cancer cells are not flexible, but our bodies are flexible, and we can actually eliminate these tumor cells gradually by taking the full power of the health ability of our body and marginalizing these tumor cells out of existence. So you're right. We need to know about how our bodies work. What does it mean to be in metabolic homeostasis? What do we do in these? How does our body maintain metabolic homeostasis in different zones of the glucose ketone index? And this is, we're working with Greg Glassman, former owner of the CrossFit, Emily Coomer Kaplan. We're going to build these MetFix boxes where we're going to be able to train people to know how to use the glucose ketone index monitor to put themselves into different zones, switching from the zones that you have mentioned, to make our lives more meaningful. But knowing that we can, by moving in and out of these zones, we can keep ourselves at a much higher state of health, and we would have it on our cell phones so that we can then monitor on the cell phone at any given time what your GKI is, and knowing what zone you're in relatively on that particular day, and how long you want to stay in these dangerous zones and how long you want to stay in the super healthy zones becomes a personal choice.
So if, to the extent that broken mitochondrial metabolism underlies cancer, because if you can't use oxidative phosphorylation effectively, you're going to use these more evolutionarily primitive forms of energy production like glycolysis. You're saying cancer cells exhibit that more. They're not using oxidative phosphorylation as much. To the extent that's true, it begs the question of what is breaking mitochondrial metabolism and the ability to use oxidative phosphorylation to begin with. Do we know what some of the major drivers are there for that disruption?
Yeah, we know most of them, and this was Albert Szent-György's oncogenic paradox, which we solved which we've already solved. He was always mysterious. He was always like, "Whoa, almost anything you've identified can cause cancer at one form or another. What's the common pathophysiological mechanism leading to chronic disruption of oxidative phosphorylation?" So when I did in my book in 2012, I went back and looked at chemical carcinogens. And a lot of these chemical carcinogens cause autofluorescence in the mitochondria, leading to disruption of oxidative phosphorylation. Oncogenic viruses, like papillomavirus, hepatitis C virus, these viruses either enter the mitochondria themselves, or the products produced in the nucleus go to the mitochondria to disrupt oxidative phosphorylation. Radiation, we know. Intermittent hypoxia, we know. Rare chronic inflammation, C-reactive protein, elevations of C-reactive protein linked to systemic inflammation. Systemic inflammation chronically damages the efficiency of oxidative phosphorylation. Rare inherited mutations, like people say, "Oh, what about BRCA1? What about what about Pete Relief-Raumeni? What about the retinoblastoma?" These are known genetic risk factors. Every one of those risk factors damages oxidative phosphorylation in one way or another. So and none of them are 100% penetrant, meaning they're secondary risk factors. Why some woman with a BRCA1 mutation gets cancer and another one with the same mutation never gets cancer. So because only the ones that have cancer have oxidative phosphorylation dysfunction coupled to a secondary fermentation.
So what you're saying is the reason that those BRCA mutations famously link to breast cancer is not because 100% of the breast cancers require those mutations. It's because if you have the mutation, it indicates you definitely have that mitochondrial dysfunction. But even if you don't have that mutation, you can still have mitochondrial dysfunction, which is why you observe both the mutation and the absence of the mutation with breast cancer.
Yes, and also 50% of the women that have the mutation uh, get breast cancer, but 50% of them that have the mutation do not get breast cancer. So why do the women with have the mutation don't get breast cancer? It seems that in those women, the mutation was never capable of damaging oxidative phosphorylation. Even though the mutation was there, OXPHOS continued to function effectively in the presence of the mutation. People say, well, what could cause that? Well, in genetics, we know what we call suppressor genes. These are other genes in the genome that suppress the action of one gene. So how does suppressor genes work to reduce penetrance of some of these inherited risk factors? And the answer is we don't know precisely how that works, but certainly an area of very interesting study. Why should somebody with a leaf-round many? Leaf-round many is the most penetrant of all the inherited risk factors, and it codes for cytochrome synthesis of cytochrome C2, which affects the electron transport chain. That's about 90% penetrant. Well, what explains the 10% that have the mutation that don't get any cancers? Maybe a suppressor gene or something. Also, it allows us to now know that if this is the case, we can use diet and lifestyle to reduce the risk of even the inherited risk factors. So we have more control. Bottom line is, once you know cancer is a mitochondrial metabolic disease, you have a much greater control as to whether or not you're going to get a cancer.
So there's many different ways to break mitochondrial metabolism. That's part of what you were just saying. Age is a factor, inflammation, pathogens, hypoxia, you name it. There's chemical exposures can break. All of these things can break mitochondrial metabolism, even though they're very different things. Yeah. Another thing I want to mention here is, as we alluded to earlier, the mitochondria are endosymbiotic in origin. They used to be free-living cyanobacteria, but that got engulfed somehow by our cells. And they are, because of their origin in that way, they are light sensitive. So they're modulated by light. And obviously, one of the ways the physical environment is very different today than it was for our Paleolithic ancestors is that the light environment is very different. The pattern and intensity of different wavelengths of light that we're exposed to are different, and the mitochondria are light sensitive. What do we know about light and its ability to drive messed up mitochondrial metabolism?
Well, that might come through the melanin, melatonin, these kinds of light-sensitive hormones. Yes, we evolved in a light-dark cycle. Light is extremely important for the normal circadian rhythms. Normal circadian rhythms maintain mitochondrial function. So if we're exposed to having less exposure to light than our ancestors did, it's just one more of the stress factors that contribute to mitochondrial oxidative phosphorylation dysfunction. Yes, a very important. It links to the circadian rhythms. We say today, what's responsible for the cancer epidemic? We have 618,000 people are predicted to die in 2025 from cancer, which comes down to 1,700 people a day. Or 70 people per hour are expected to die this year from cancer. Clearly, we're in an epidemic of unhealth. Okay, so you have light sensitivity. You have a lot of highly processed carbohydrates that are minimally nutritionally of value that create systemic inflammation. You have lack of exercise. You have emotional stress. Whether you're sitting in traffic, you're looking at the computer, you're looking at your cell phone all day long. You have poor sleep as the result of the circadian rhythm disruption. You put all those factors together, and you have an epidemic, not only of cancer, but of all these chronic diseases as well. Type 2 diabetes. You have obesity. You have cardiovascular disease. You have cancer. You have the whole package, dementia. You put all those things together, and the majority of our chronic diseases, including cancer, are the result of mitochondrial dysfunction coupled to a substrate level phosphorylation contributed by a whole variety of different factors in the environment. This is called the oncogenic paradox. That's why we solve this. And then you can use a glucose ketone index to bring yourself back into these healthier zones. So do what you need to do to get light, to reduce your stress, to get more exercise, and you bring yourself back into a more normal zone, thereby reducing cancer risk and chronic disease risk. So clearly we have a clear strategy. The question is, can the American public recognize this and now take steps to prevent cancer? And if you do get cancer, how to manage it and prevent other chronic diseases?
I want to ask you some more details about therapeutic ketogenic diets, such as those that you and others have used in this area of research. So there's many different ketogenic diets one could eat. And what I mean by that is, the fatty acid profile of your ketogenic diet could be very different from someone else's. You might be having high saturated fat, high polyunsaturated fat, monounsaturated, all the different combinations thereof. We mentioned earlier that polyunsaturated fatty acids, they can be oxidized readily by reactive oxygen species, and this produces toxic byproducts, reactive aldehydes and things like this that damage cell components. And we alluded to some other work that's been done related to that stuff. When you guys use a therapeutic ketogenic diet, what are the specific fatty acid profiles that you use, and what's important to think about here in terms of fatty acid composition from saturated to more unsaturated fats?
Well, the saturated fats, medium chain triglycerides are more provocative for building nice ketone bodies than polyunsaturated fatty acids. Polyunsaturated fatty acids can give flexibility to membranes like olive oils and things like this. I mean, there's a number of polyunsaturated fatty acids that are very fish oils and things like this that are very healthy for you. But to make ketones, and the answer is we use medium chain triglycerides, avocados. We try to do it with as much natural fatty acids as possible, always calorie restricting them, always using the glucose ketone index, which is the ratio of circulating ketones to glucose, as kind of a guide. People say we use the GKI more to know what zone you're in, as opposed to the types of fatty acids that you are consuming. But we know there are natural ways to get ketones, ketogenesis, natural ketosis. You're mobilizing the fats predominantly out of your adipocytes. But at the same time, if you take in a diet with medium chain triglycerides, whether it comes from a plant or an animal, you'll produce better all natural ketone bodies. And again, it should be done under low glucose, because the lower the glucose, the more the need for the body to make ketones from the liver. So, and you're right about that. I mean, we're experimenting more with that. What are the best diets to produce the most natural forms of ketone bodies? Again, with understanding calorie restriction and using the GKI as a quantitative measure. So we no longer need to be in a mysterious state. Oh, I think I'm in ketosis by pissing on some strip or blowing out something. We actually have a ratio now that is based on blood. We have a glucose continuous monitor. Now we're going to eventually we'll have glucose ketone continuous monitors. So we'll be able to know precisely what level of ketosis we're in and the types of fatty acids that we can consume to bring us into these zones in a healthy state.
Okay, but it sounds like in essence what you're saying is that, okay, the ketogenic diet is a high fat, ultra low carb diet. You're going to use those fats to make ketones for energy as opposed to glucose. But those ketones, those fatty acids that you're using for energy, those are more readily made from shorter chain, smaller fatty acids, whereas the longer chain polyunsaturated fatty acids are more likely to be used for like membrane structures and things like that.
Yeah, that's true. They're not burned as readily as the other fatty acids. But some of them are unhealthy, like you mentioned. They can create inflammation, like arachidonic acid, 24. That's a provocative inflammatory fatty acid used for inflammatory conditions, where others like omega-3s are used for anti-inflammatory activities.
So it sounds like you would not recommend someone to have a very high omega-6 polyunsaturated fatty acid content in their ketogenic diet.
Yeah, I think we need to work better on that. I think we need to be more cognizant of the foods. Ultimately, we're going to be taking in the food. We want to have a food menu that is palatable and therapeutic at the same time. And that's where food science and technology will start making their move into this space, into this zone, improving, making natural ketone bodies more efficiently from foods that are known to be healthy,
Hmm.
as opposed to those that are not being healthy. And I think that's an area that will be exploding here in the next several years, and we're going to have the quantitative biomarkers to let people know. Number one, how do you feel? What state are you in? Oh, it's like unbelievable. So, and it might be different from one person to another. We can't group. We have people that are different in age, in gender, in culture, in religion. All of these will have to be recognized when we're building these new strategies for managing chronic disease. So this is the future. Right now, you just have to have the platform, the bedrock upon which to build all these in a logical way. Recognizing that cancer is a mitochondrial metabolic disorder now allows us to shift to logical, science-based strategies for managing this. And of course, the chronic disease world will be dragged along with this, because the cancer is recognized as the number one fearful disease in the world. It's not getting better; it's getting worse. When someone is diagnosed with diabetes, they don't feel like the angel of death is looking over their shoulder. When you get cancer, you're looking at the grim reaper. Well, this has all got to change. Yes, it'll be an unfortunate diagnosis, but we have a clear strategy on how to manage that without collateral toxicity. There should be none of these medieval treatments of poisonous chemicals, surgical mutilations, the way we're doing it, and incinerating radiations. I mean, we can use all of those strategies, but now they have to be recognized to be used in a very different plan than they are currently being used. So I'm very hopeful for the future of managing not only cancer, but multiple other chronic diseases, and giving individuals the tool to know they'll now be able to take much greater charge over their existence, because they'll have the definite tools and know what to do and how to do it. And that's a personal choice. You can either do it or not do it. We're not forcing anybody. We're not going to tell anybody, don't eat that, don't eat this. You just look at the GKI monitor and tell you whether you should eat it or not. You make the choice for yourself.
Hmm. Um, I want to zoom out a little bit. So cancer at the level of society, where are we with cancer in terms of, how common it is and whether or not certain cancers are becoming more common today than they used to be? Can you give us a general sense of that?
Yeah, the general sense is that it's becoming more and more in younger people. I think I'm not sure we're seeing it. I'm seeing it. I get calls and emails all the time from young people asking me to help them with transitioning to metabolic therapy, and we give them a plan. We've written a very comprehensive treatment protocol for managing glioblastoma, which could be used for any cancer. But we're seeing so many people in the late 20s and early 30s now with colon cancer, breast cancer, various types of cancers, where we would never see that many in the past. And as I said, when this new big paper we put out on the Warburg hypothesis and the emergence of the mitochondrial metabolic theory, we have a big section in there where we've now incorporated forever chemicals and microplastics and these kinds of things that are now entering our environment in greater amounts than we had in the past. You couple that with an obesity epidemic where chronic disease C-reactive protein is now elevated, and you combine that with these other things, and I think that could account in large part for why we're seeing cancer at younger and younger ages in these people. So everything is related to our metabolic homeostasis and the exposures that we're getting for provocative agents that will chronically disrupt oxidative phosphorylation.
When we go back and we think about the sociology and the history of science piece of this that we were talking about towards the beginning, it reminded me of the saga of the diet hard hypothesis and Ansel Keys that many have talked about in the diet world, where, long story short, my interpretation of all of that that I share with many people is that, Ansel Keys's ideas became dominant not because they won a scientific battle on a level playing field, but largely because of social and political influence. The right people in the right positions championed those ideas. They suppressed alternative ideas that they were competing against through artificial means. And those ideas became enshrined at the level of public policy, such that for decades at a time, the food pyramid and all that stuff was constructed with this one model being treated as unassailable fact, when in fact it was just one model that never had a total true consensus behind it. And I couldn't help but notice that in the Warburg cancer biology saga that you told us about, something similar happened where the ideas weren't competing head to head on a pure scientific basis, but they sort of moved and became more prominent for social and political reasons.
Yeah, I think so. But this is the nature of the way we move forward. And as I said, during the Copernican revolution, that was not taken lightly by the Catholic Church and other religious organizations, and led to a lot of upheaval in the way people and the way establishments viewed their position in the world. And I think the paradigm shift for cancer, going from a genetic disease to a mitochondrial metabolic disease, will have every bit of tumultuous change as did the Copernican revolution. Because if in those days it wasn't, if you thought the sun was the center of the solar system or the Earth, whether you agreed or disagreed, it wasn't associated with 1,700 people a day dying. Our misunderstanding of this situation today, that cancer is a genetic disease, is absolutely responsible for the 1,700 people a day dying. Those people are dying because they're being viewed as having something other than what they have. They do not have a genetic disease, so these precision medicines that are all based on the somatic mutation theory are indirectly allowing the field not to move forward. And as the result, we're getting all these poor people that are dying. So the consequences of not knowing what you're talking about scientifically have dire consequences for people. The suffering and death is enormous that we're seeing with this cancer today. And that's all because they refuse to accept the Warburgian, the idea that this is a mitochondrial metabolic disease. And revenue generation is more important than patient outcome. We know this for a fact. The National Cancer Institute says it's a genetic disease, funding millions of dollars in grant support on a flawed theory. So we're getting more and more dead people based on a lack of knowledge or ideological dogma that's preventing the transition, and this is tragic, a tragedy of greatest proportion.
Mm-hmm. So, with your view of mitochondrial metabolism taking center stage in terms of cancer, what are some of the best and simplest ways people can manage their lives to prevent cancer? Would you say that people should be going into ketosis one way or another, at least intermittently, as part of their strategy?
I think so. And exercise plays all we're doing is bringing ourselves back occasionally to the Paleolithic period where cancer was not prevalent. How do we know that? From the work of Albert Schweitzer, looking at tribes of people that live according to their traditional ways. So yes, I agree. I think that as long as we are aware of what we need to do and how to do it, then it becomes our job to act upon that. But right now, the misinformation associated with the food pyramids and with what cancer is, this level of misinformation is causing a tragic consequence to the health and vitality of our population. So yeah, we can prevent cancer if you can keep your mitochondria healthy. It's very hard to get cancer when mitochondria are healthy because mitochondrial dysfunction is the origin of cancer. So maintaining metabolic homeostasis is absolutely essential for preventing cancer and other chronic diseases. So it's not bad luck. It's not this crazy nonsense that you hear about people talking about. It's our diet and lifestyle issues that predominantly are responsible for our health. But at least if we have tools to put us into zones that we know about, then it becomes up to us that we have greater control over our destiny.
Anything you want to reiterate or any final thoughts you want to leave people with about what we discussed around cancer and metabolism?
No, I think you've done a wonderful job getting down into the nitty gritty. Most folks that I talk about on these podcasts, we rarely have an opportunity to go down into a deeper level of our understanding. You have to realize that in our lab here at Boston College, this is all we do. We discuss these things day and night, night and day. We have groups here. All we do is sit around doing experiments, testing our ideas, and discussing the concepts. And we always looking for somebody who's going to come at us with some piece of evidence not consistent. And we look for that's why we're offering $100. You show me a cancer cell that can say that, I'll be more than happy to give you the $100.
$100, people.
Yeah, $100. I'll give you some of my friends will give you $1,000. But the issue is here, we haven't had any takers. And with those who say that, we always find that you got plenty of fermentable fuels in your environment. Get rid of that and see what happens. But we're testing all this. And the other thing too is our research is supported predominantly by private foundations and philanthropy. So we do what we think we need to do and how to do it ourselves. And you'd be surprised. This is a revolution coming. This is a health revolution that's on its way. And it's going to be restructured and it's going to be open to the people and they're going to know how to use this. And the better that we have the best animal models of cancer, all natural, none of this artificial, these genetically constructed monstrosities, patient-derived xenografts, all these crazy things that think that you can get grants on but have very little relevance to the real world. So we always do everything naturally. We have natural metastatic cancer. We know how metastatic cancer works. We know how to stop metastatic cancer. We know how to do all this because we have the science behind us to do all this stuff. And as long as we get donations from people who want to be part of the movement, the transition, the paradigm shift, whatever you want to call it, they're excited about this. The future is excited about this. So we're moving in the right direction. And private foundations and philanthropy are giving us the support that we can do all this. And I keep publishing these papers in open access journals so everybody can read the papers. People ask me all these questions. The answers are in the papers. Please read the papers and you'll get the answers.
All right. Dr. Thomas Seyfried, thank you very much for your time.
Well, thank you very much.
Visit mindandmatter.substack.com to find all of my content. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.